You are listening to the Pet Radio Show on PetRadioShow.com, a Pet Radio Show podcast with Robert Paul Hudson, your host. It is my pleasure to introduce Dr. Jeffrey Bryan, Professor of Oncology at MU College of Veterinary Medicine, and author of a study to develop a vaccine against a type of bone cancer in dogs and opening a door for human research. Welcome to the Pet Radio Show, Dr. Bryan.
Thank you very much. I'm glad to be here.
Now, could you begin by just kind of giving us a little synopsis of the scope of this study and what it means to the dog owner and to us people?
Yes, absolutely. This is a technology that was developed by Dr. Gary Wood, an immunologist and scientist at Elias Animal Health in Kansas City, that uses the patient's own tumor cells to form a vaccine to cause an immune reaction that is then supported by what's referred to as an activated T-cell infusion following that. So what happens with these patients is if a dog comes to us with bone cancer and was participating in this trial, they would be evaluated thoroughly to make sure they did not already have spread of the disease or metastasis. And then when we were sure they did not, we would send them to surgery to have an amputation performed, and part of the tumor would be collected after the amputation to be sent to Elias Animal Health in Kansas City to make a vaccine. The dog would then be vaccinated 3 times with its own cells against the tumor, and the purpose of that is to get the immune system to recognize the mutated proteins in the cells as something that it should pay attention to and eliminate. Much like viruses or bacteria hide in cells and cause viral or bacterial proteins to be made, which lets the immune system recognize those cells and then eliminate them because they're harboring a dangerous enemy, the production of mutated proteins in cancer cells gives the same kind of a signal to the immune system. But cancers are very tricky and aggressive at reducing that immune response to protect themselves against the body's own immune system. So by vaccinating, we get the body to pay attention to these abnormal proteins. That makes lymphocytes, which are immune cells, be produced that recognize those proteins, and those circulate in the blood. We then harvested those from the blood through a process called apheresis. Anyone who has donated platelets knows what it's like to undergo apheresis. It basically takes some of the blood out of the body through a catheter, puts it through a centrifuge to separate the different types of cells, and then the apheresis machine keeps whatever types of cells you tell it to. And in this case, we're harvesting the lymphocytes from the blood. Those go back to Kansas City. They are expanded in cell factories to large numbers, and within those large numbers are these tumor-recognizing lymphocytes, and then they're activated to be very aggressively pursue their target inside the body. And so they're sent back to the hospital and are given like a blood transfusion would be given over about an hour, and then after that, we trust the immune system to start attacking those cancer cells and hopefully eliminating them. And we did this in a study. The pilot study contained 15 dogs, 10 of which got the entire treatment. And of the dogs who got the entire treatment, about half of them remain alive to this day, more than 18 months out from the date of their treatment. And that's a very encouraging result because usually we see that dogs only live an average of 9 to 12 months after surgery and chemotherapy. So in this case, these dogs got no chemotherapy at all, and so far appear to be doing better than if they had gotten chemotherapy instead of immunotherapy. So we're excited about that.
So it gives the animal an extended period of life, but it's not a cure either, though, right?
We are not calling it a cure because it's early days. However, the dogs that have gotten this, the 5 of the 10 who got the whole thing, still have no recurrence of disease at all in their body. And we do know that of the dogs who are treated for osteosarcoma, a small percentage will never relapse again. And so those you would consider to be cured. Usually that's about 10% of all the cases that we see go on to live very long lives and don't have recurrence of their cancer. In this case, half the dogs that got the whole treatment have, and that is very exciting.
Now, why did you pick this particular type of cancer, or bone cancer in particular, to do this study? I mean, would this be a viable study for any type of cancer?
That is a great question, and we would say that yes, this potentially could be an approach that could work for almost any type of cancer. We know in the immunotherapies that have been developed for humans, the types of cancers that create the most mutations and therefore the most abnormal proteins seem to be the ones that respond best to the immunotherapy. And so I suppose there is a chance that among all the types of cancers, there are some that would not be as well controlled by this approach as others. But in the case of bone cancer, we know it generates a lot of mutations. That's something that our own group has shown through some genomic sequencing, and others have shown the same thing. It's a disease that is very aggressive, and the chemotherapy has not been a cure for very many dogs, and so looking for other treatment options is very reasonable. And it's also a disease that we see a lot of. Unfortunately, dogs get bone cancer at a rate greater than 10 times that of humans, and so it's very common among dogs. And so we're always looking for ways to control this disease better. And so the immunotherapy was a very natural extension into this particular cancer type. And we're very excited about the results. We definitely would like to see this generalized against more cancers. Right now, it's being offered by the company as a commercial application for bone cancer specifically, and so we're hoping that as the numbers continue to increase of dogs that were treated with this, the great news will continue, and it will control disease in a larger population as well as it has in this initial study.
Now, what does this mean for us humans and treating cancer in people?
That's the million-dollar question, literally, on the human side. I don't think anybody is unaware of the excitement that human medicine has right now about immunotherapy. It's dominated the news. There are headlines every day of new immunotherapy drugs. The problems with the immunotherapy drugs is they have been extremely expensive. They have not been broadly effective for every patient getting them, and there have been cancers that have not been particularly effective for at all. And so we're always looking for new ways to apply immunotherapy. This activated T-cell therapy technique is something that's relatively accessible to humans in a large scale, and so that's very appealing. Dr. Wood has conducted studies in humans with brain cancers in the past, and there was evidence that it was beneficial to many of those patients. So I do think there's a lot of reason to think that this will help. The challenge with osteosarcoma in humans is that it tends to occur quite infrequently, only 600 to 800 cases a year across the United States, and it tends to occur in children. And so the challenge will be identifying how you would conduct a clinical trial in this group of patients and who would still likely benefit even if they had already failed first-line therapy, which is often when these kinds of trials occur. And so as we understand this better in dogs and we start to understand better why dogs respond or fail in spite of this therapy, then hopefully we can work out some of the processes to make it more effective and to identify potential failures ahead of time before they get the therapy so that you could apply those same metrics to kids who are considering going through such a clinical trial to hopefully have the best outcome for anyone entering the trial possible.
I see. Oh, how interesting. At least in some circles, there's some controversy about vaccinations as a whole, whether we're over-vaccinating or whether they're really effective or what. How does this fit into all that?
That's a huge point, and in fact, some of my collaborators that work with us in immunotherapy here at the university and I were discussing exactly that last week. One of the big problems with the vaccine controversy against infectious disease right now is that a lot of that was amplified and driven by what turned out to be a completely false link between vaccination and autism. There was a paper that described the link, and it turned out that paper was entirely falsified and has been fully retracted affected. There was no truth to it at all, but it became so ingrained in popular culture that I think people don't remember that it was not at all true. And so there are people who are concerned that vaccines are causing harm that has not been fully documented. There's also a very small subset of people and of animals that do get harmed by vaccine, and the Vaccine Indemnity Fund covers people who have been through that. So there's no doubt that any medical therapy has some risk, but I think as Washington State is seeing with the sudden outbreak of measles within the state for the first time in many decades, there are reasons we need to keep the herd immunity up and protect ourselves, and especially protect the immunosuppressed people among us against these diseases. This vaccine approach is truly a vaccine in the technical sense of what it is, but it's not really a preventative health vaccine. So, for example, you wouldn't consider vaccinating a dog who didn't have cancer with another dog's cells to try to prevent them from getting bone cancer. This is an immunotherapy treatment for dogs who have already developed cancer. And the interesting part of this is that these vaccines made from their own cells appear to be nearly innocuous. And in fact, they have been, this approach has been tested for many cancers over many years and has not been particularly effective unless it's augmented by something else. So I think the real key in this case is the activated T cell therapy that we give, and then we treat them with an immune hormone called interleukin-2 for 5 treatments after they get that activated T cell vaccine or that activated T cell infusion to keep the immune reaction going and keep the cells looking for those cancer cells to eliminate them. So I think that the real target of this is going to, in almost every case, be the cancer. And so I think some of the concerns that people have about the general vaccine approach for preventing disease aren't very applicable in this population.
So how far off are we from Mr. Smith being able to go down to his vet office to bring his dog and to get him vaccinated for this?
If a dog had osteosarcoma today, they could consider having this done. The company is able to offer this on a commercial basis. It is still considered experimental by the USDA, but the USDA is aware of the company's efforts and have approved that they move forward offering it on a commercial basis. The company is working towards full USDA approval for this process. But what would need to happen is that the dog would need to have the tumor still in place. It can't work after the limb has been amputated. You've got to be able to get to the tumor cells immediately post-surgery while they're still alive and well and can be made into a vaccine. And they need to live close enough to one of the apheresis centers that the company sponsors, like the University of Missouri, the University of Kansas State University, the University of California at Davis, either up in Davis or down in San Diego, and they have other locations on the East Coast and in the Northwest that would allow the apheresis to happen. Beyond that, they need a veterinarian who's willing to help them collect the cells for the vaccine, to administer the vaccines after the company makes them, and then to administer both the T-cell infusion and then just the subcutaneous hormone injections afterwards, all of which is relatively straightforward and well described. So really, any dog that develops osteosarcoma today could be eligible for this and could get this treatment if the family wanted to pursue it.
But you can't just walk into any veterinary office and get the vaccine. So we're a ways off from that happening.
Right. The veterinarians would need to communicate with the company and make sure that they can perform the procedures that the company needs done. And so if the veterinarian was willing, it is possible that they could do it. Certainly most specialty hospitals could allow it. So if they can travel any distance at all, they probably can find someone to help them get this.
I imagine this, with all that's involved, especially trying to track down, and coordinate the whole thing, I imagine it's a little pricey.
It is. As it stands now, that price point is probably about $10,000, including the surgery and the preparation for vaccines and everything else. To put that in perspective, here in the Midwest, chemotherapy is probably $5,000 to $7,000 in most practices, especially most specialty practices, and surgery and chemotherapy is probably $10,000 on the coasts. And so, as expensive as that sounds, I think that the value for what it is substantial.
Well, it sounds as if your study was deemed a success. Then, I mean, you're already past the trials, and it's out there to be ready to be implemented.
We're very excited about that. It's one of the first concepts that I have been involved with, from the design of the clinical trial all the way to the availability of a commercial product, and I find that extremely gratifying for sure. We are writing up the trial. We'll submit it for peer review and publication so that everyone can judge our methods for themselves, but we are very confident in what this outcome is telling us.
Now, was there anything in the course of this study that surprised you? Were there any results that you weren't expecting?
I would say that the outcome is superior to what I would have allowed myself to hope for. This has been such a dismal disease. It's so aggressive. To give you a comparison, we have been involved in a number of osteosarcoma trials involving surgery and chemotherapy in the last few years, and of 30 dogs that were enrolled in another pair of trials, only 3 remain alive after a long period of time. And so to have half of these dogs that we treated with this approach still be alive is very rewarding and very exciting. We did see evidence that without proper pre-medication, the immune system can definitely make you feel crummy when it's reacting to something strongly, and so we worked with the company to identify medications to give along with the T cells to make sure the patients stay comfortable. And I think that the overall tolerability of this was also a very pleasant surprise, that the dogs tended to feel well, tended to respond well to the treatments, and overall did very well throughout the whole course of their therapy. It has been tested in a number of other different ways, not as often through this disease, and certainly not quite this elegantly. And so that's where I think that the immunology knowledge that Dr. Wood brought to the problem was just critical to understanding what the main steps would need to be to really optimize this. And I expect that over time we will continue to optimize protocols and continue to change things as we go. But this certainly turned out to be a very fruitful protocol in this iteration, and we're excited about that.
Well, Dr. Jeffrey Bryan, professor of oncology at MU College of Veterinary Medicine, thank you so much for coming on here and telling us about this.